It's Libby, and I'm going to be talking about microscopy in biology today. So, when using microscopes, two variables matter the most: magnification and resolution. Magnification tells us how many times larger an image seen through a microscope is compared to the real object. And resolution is the ability to distinguish between or tell apart two or more objects that are close together. So when using microscopes, what are the two variables that matter the most? So we know that these are magnification and resolution. So for microscopes, resolution and magnification matter the most. Magnification tells us how many times larger an image obtained using a microscope is relative to the real object. And to recap resolution, this is the ability to distinguish between two or more objects that are close together. So, an example of magnification is that a cell has a diameter of 0.001 centimeters. An image of that cell seen through the microscope has a diameter of 5 centimeters. So the magnification is the relationship between the size of the image seen with the microscope, here, which is 5 centimeters, and the size of the real object, 0.001 centimeters. We work out the magnification by dividing the real image, 5 centimeters, divided by the real object, 0.001 centimeters, to get times 5,000. So to recap that equation, magnification is the image size divided by the actual size of the object which is being magnified. So to look at that equation again, magnification equals size of image divided by size of the real object. So if we are looking at an object which is 2 micrometers, we want to consider what sort of size that could also be considered to be the same as. So this is the same as 0.00002 meters, or 2 times 10 to the power of minus 6 meters, or 2 micrometers. If you want to know more about converting between different distances or lengths, I'd recommend listening to the episode on standard form in this topic. So resolution: if two spots are very close together, a low resolution microscope may not see them as separate objects, as they sort of blur together. Using a microscope with a higher resolution will allow them to be seen as 2 separate objects. So to look back at our magnification equation, if a cell has a diameter of 0.001 centimeters and an image of the cell viewed through the microscope has a diameter of 5 centimeters, we know from our equation that magnification is the image size divided by the real object. So 5 centimeters divided by 0.01 centimeter, which is equals to a magnification of times 5,000. So what is a magnification of times 240,000 in standard form? So if we want to look at how many decimal points we need to convert in order to have it to standard form, you would want it to be, 1, 2, 3, 4, 5, to make it 2.4 times 10 to the power of 5 is equivalent to times 240,000 magnification. So to work through an example, let's say an object is 5 millimeters long, which looks 10 centimeters long when we use a microscope. So we know that its actual size is 5 millimeters and its image size is 10 centimeters. And we want to work out what is the magnification. So the first step is to remember the equation for magnification, which is the magnified size of the object, or the image size, divided by the real size of the object. So then we remember to convert one of the lengths so they're all in the same unit, as at the moment we're looking at an object of 5 millimeters and an image size of 10 centimeters. So we're going to convert the 10 centimeter image into millimeters. And we know that 1 millimeter is equal to 0.001 meter. So 10 centimeters is the equivalent to 100 millimeters. Then we substitute these values into the equation. So we will know that the magnified size of the object is 100 millimeters. We know that the actual object is 5 millimeters. And so 100 divided by 5 is 20. So the magnification is times 20. So to recap the two important factors for magnification, we know that resolution is the ability to distinguish between two or more objects. Okay, so that summarizes microscopy, the first episode for now. Please move on to the second episode if you'd like to know more.